Hi everyone. Hallo zusammen. We are live for the first episode of Next Gen Scientist at the Institute of Science and Technology, Austria. I am Ana Rita Cavaco. I am your host, and I am excited to bring you the amazing science done here at ISTA and the stories of the people who do it. My first guest is Carolina Camelo. She is a developmental biologist. She is a postdoc researcher at let me check, at the Morphogenesis in Development lab with Professor Eisenberg, and you will tell us all about becoming a developmental biologist. If you have questions for Carolina along this episode, feel free to ask them through the comments, and we will address them in the Q&A session. Let me just activate the chat. So feel free to ask your questions and we will answer them in the end. So Carolina, thank you for being here today. Hi, thank you for accepting to be our first guest. Tell us a bit about your work here at ISTA. What do you do as a developmental biologist?
Okay, hello everyone. Hello Rita. Thank you for inviting me to be your first guest in your new podcast. So as you said, I work as a developmental biologist, and I work with the sea squirts. This is a marine species that can be found in the bottom of the sea, and we work with the embryos of this species, which means we look at the one. It's still an egg and the sperm. We look at the fertilization, and we look at the first developmental step. So for my specific project, I work until the 16 cell stage. So I look at everything that's between 2 cells and 16 cells. And I'm particularly interested in how the shape of these embryos is regulated. So why when they are 4 cells, they look like squares. When they are 8 cells, they look like cubes. And I look at how these shapes come into place.
Okay. A few days ago, we met in your lab, and we prepared a small activity where you showed how to produce a sea squirt embryo and how to look at the first stages of their development. And later on, when we publish this episode, you can also see this activity that we did together. How is understanding this process of embryo formation and its first stages, how is understanding this important for your work?
So what I showed you and what you can see later in the video is basically all how my experiments start, actually. So we usually have an animal, and we have to sacrifice the animal to recover the eggs and the sperm. And then what we did in the video was to do a fertilization assay. So we put the eggs together with the sperm, and then we let it develop for the first 2.5 hours, more or less. And this is basically how 99% of my experiments start. We look at how the embryo is developing. We can label different things in the cells. In the case of the video, we labeled the mitochondria, so one of the cellular organelles, and the plasma membrane. But we can also label specific proteins that I'm interested in, for example. For most of my project, for example, I label proteins that are at the contacts between cells, and then I can analyze how much protein is there, if the protein is distributed equally in all the cell-cell contacts, or if it's more in one side of the embryo, and so on. For example, I can also manipulate the shape of these embryos. We can do that, for example, by putting them. You will see in the video that they are in these round wells while we do the imaging for this specific experiment. But we could also, for example, put them in a triangular well, and then we manipulate the shape of this embryo, and then I look again at these proteins. I see how they are different from the normal embryos that grow in normal wells, let's
Okay,
say.
so basically you can make them grow in many different shapes.
Exactly, exactly. So I can then basically the idea is to look at how these molecules, how these proteins, how these cellular components um, look like in controls, in normal wild type embryos, and how they look like when I change their shape, so that I can understand basically the relationship between the biochemistry, so the molecules, the proteins, and the shapes that arise from these.
Very cool. But in your work, in your daily work, you do many different things other than this. So, how does a typical day look like for you?
Yeah, so I do a lot of microscopy. This is one type of microscopy. Sometimes I do, of course, different ones. Um, my normal day starts with opening C squirt and doing this fertilization assay. Depending on what I want, I can use these markers and directly go to the microscope. They are fluorescent markers we put in the media, as you see in the movie. Then we go directly to the microscope and image this for 2-3 hours. In other experiments, I also, for example, can inject these embryos. And then in this case, that day, I will not go directly to the microscope. I will just prepare the eggs and the things for the next day. So I inject and then I wait for the next day. And then also a lot of the work includes image analysis. So we receive all these data from the microscope, and then we use some image software to process the data, to recover the amounts of proteins we have in the cells, how the shape parameters, all these kind of things. So it's basically going to the microscope, doing some experiments, and then analyzing this in the computer.
Sure, and then to later on have a nice story to publish as well.
Yes, of course. Yes. So of course the idea is then to come up with some conclusions and then eventually publish this in a story in a paper, and yeah,
Yeah.
there's still something. So some time until then. But
Yeah.
yes.
Now, obviously, to do what you currently do, you needed to become an expert in developmental biology. When did you realize that you wanted to become a developmental biologist?
Um, mm. I think so. I initially studied biochemistry because I was not so convinced about biology per se. I really liked chemistry, so I wanted to have some more knowledge on the molecules, not so much on the animal itself or the plant. But then at the end of my bachelor, I did my bachelor exactly at the end of my bachelor degree. I took an extra course on developmental biology, and I really, really liked it. I think it's because you look at how things develop life, and that is very fascinating. Like you can just see the fertilization, and you can see how the embryo is developing, and it's very dynamic and it's very catchy, at least for me. And you get to get very nice images and movies. And I think that was the first time I started thinking about more developmental biology. And then for my PhD, I already did developmental biology, although I was looking more into cell biology. It was just that it was within the development, but I was not looking at development per se. And but then for my postdoc, I decided to go more into developmental biology and biophysics.
I see. And growing up, you already said that you started by biochemistry because you also wanted to understand what was happening at the molecular level. But growing up, did you always like biology? How did you feel about science?
I think I always liked biology or science in general because when I was very small, I really liked to watch National Geographic documentaries. I remember me and my brother and my sister, we always had the fight for the controller of the TV because she wanted to watch cartoons and I wanted to watch documentaries, and always wildlife. I always wanted to watch wildlife documentaries. And yeah, I think it was already there, my passion for biology. But then in school, I didn't like it so much to study like the organs and all these things. And I really like chemistry, but I also found it very dry. That
Mhm,
like
mhm.
I wanted to put the chemistry in some more context, and that's how I just came across the name biochemistry, and I thought, oh, what is this actually? And then I started looking more into what exactly it means to be a biochemist and what you would learn at the university. And then I decided to apply because I thought it fits better to these two things I like in chemistry and biology, but not themselves like separate.
Yeah. So, you already uncovered a bit of what I'm about to ask you just now, but to be at the point where you are today, uh, what was your path? Tell us a bit about all the cycles that you did, what you studied.
Mhm. Yeah, so as I told you, I did a biochemistry bachelor in Lisbon. I'm from Portugal, and I studied in Lisbon for my bachelor and for my master degree, which I also did in biochemistry. And then I decided to go abroad, and I did my PhD in Germany. And this I was working cell biology, developmental biology, in a laboratory, also working with a model organism, but in this case, fruit flies and not sea squirts. And then after my PhD in Germany, I decided to move to ISTA for my postdoc, where I am for 2 years, 2 years and a half almost now.
And while growing up, what were your aspirations? What were you hoping for? What did you dream for?
Uh, oof, that's a very tricky question. um, I think I never really expected, or I was not, I didn't grow up thinking I would become a scientist.
Mhm.
But I had a lot of fun and I really liked studying. I always liked learning new things, especially. And so it was more with this that I went to university. I just wanted to learn more things. And then when I was at the university, I had the chance to work in a laboratory. I got a small fellowship to do some undergrad research. And this was my first contact with the real lab. And I really liked it. I know that I remember that after some very tiring days in the classes or studying for exams, I sometimes was still like, oh, no, I still have to go to the lab and prepare media for my East. But then I was in the lab and I felt like, I don't know, I was happy again and I got energy again from somewhere. And this is when I thought maybe I actually really enjoy being in the lab and this is what I want to do for my life. And I think this is when I decided to be a scientist. I didn't have this aspiration before that. No.
Cool. And are there any people in your life that you consider somehow responsible for you deciding to become a scientist?
Um, not so much. I think there's no one that I really, I don't know, praise for this. When I was small, like, as again, I was always looking at documentaries, so I had a huge fixation with Jane Goodall because she was, I don't know, living with the chimps, and I thought, oof, this is an amazing life. But this was still far from reality, and at that point, I think I wanted more to do this traveling and videos with animals than exactly becoming a scientist. And then I think through my career, probably my master thesis professor or mentor because she was always very supportive of my work. She encouraged me a lot to go abroad and to try to reach, like, to go to a lab that would really benefit me and that I would really like the project. I remember I got an offer from a different lab, also in Germany, and I refused this offer because I felt it was not correct. Like, it was, I didn't feel like I would be happy in this lab. And while everybody was a bit like, "Are you crazy? You got a PhD position. You should accept." She was very supportive. She understood why I said no, and she was encouraging me to find a better place where I could do my science.
Yeah, indeed, it's very important to be in a place where you really want to be, because a PhD is a
It's, yes, it's a long
it's a
commitment.
considerable
Yeah,
commitment.
yeah,
Yes,
yeah, and it should be fun and not just a pain.
absolutely. And you found your place.
Yes, I
So,
found my place.
and to be at this moment of your career where you are today, what do you consider to be the biggest challenge that you faced?
I think when I moved to Germany, probably because I was, of course, younger back in that days, and it was the first time for me to live alone and to live abroad in a country where I didn't speak the language. And I think this was the most challenging. It was very lonely in the beginning, and yeah, it was, I had to consider really if I wanted to do this, leave my family, all my friends in Portugal. But yeah, at the end, I'm happy that I went abroad and overcame this, and now I actually enjoy living abroad.
Yeah, I can relate with that. And so, on the other side of this, what is it about your work that makes you wake up and go, that gives you energy, motivation?
I think just the job itself, that you my job is asking questions and learning new things all the time. Of course, like, you tend to forget this when you are in the lab and you do the same things over and over, but we have a flexibility to think and to ask different questions, to test new things that no one ever tested, and to find new answers that no one ever thought about it, or they could never find them, let's say. And with this, you learn new things about the animals, you learn new things about how nature was built, why do the cells behave this way. And I really like this. I really like to come and have always new questions, new hypotheses that I can test, and think about cool experiments I can do, and and do them. Yeah.
And what does it mean for you to be at this point of your career?
Uh,
You are at this moment a senior postdoc, right?
Towards
To
seniority.
Yeah,
Yes.
towards seniority. So, what does it mean for you to be in this point?
Um, I'm happy to be here. I There was a lot of moments because, of course, it has been a long process since bachelor, master. PhD is also very long, and it was. uh, I liked my PhD time, but it was also very challenging. And there were moments where I thought of giving up and doing something else. Um, and I think I'm quite proud that I said, no, maybe I want to do a different project, and maybe I want to keep in science. I want to keep going in science. It's just that it's time for me to move on and to stop this project and move to a new project, to a new country, and look for something that fits me better. And while there was these moments that I wanted to give up, I still decided to give it another try and to really like check if I really love science. And for that, I'm quite proud that I can be here now, and I'm happy that I can do this job and be here, well, for these 2 years, maybe a bit longer. And so for now, I'm quite happy to be at this place.
Very well. And what about the future? What's next for you? What are your plans?
Uh, yeah, I'm not sure yet. Uh, of course, in when we are in this position, it's always kind of a bit expected that we then the next step would be professorship. I'm not sure if it fits me. I really like to be in the lab, and being a professor means you supervise more and you do less experiments. I'm not sure yet if this is the right path for me. But if not, it's also fine. And there's many other things I like. I really like also science illustration, for example, and trying to make some schematics to make my science more understandable. This could be an option. And I also really like science policy. So trying to make to change policy so that PhDs, postdocs have a better work-life balance, that we promote women and other diversity in science. And maybe this would also be an option for the future. But for now, it's still quite open.
Yeah, because in fact, there are many things a scientist can do
Exactly.
besides being in the lab and develop a project.
Exactly. Yes. Yeah. And I think this critical mind that you develop when you are a scientist, also during the PhD, it's important for so many jobs. It doesn't mean you have to do all this PhD postdoc professor. There's a lot of ways out when you want.
That's very true. So I would like to finish this part by asking you if you could meet teenage Carolina, this girl who was so into National Geographic, who wanted to know more. Um, which advice would you give her?
Um, hmm. Yeah, this Carolina was a bit naive. I mean, I would tell her maybe to believe more in herself and sometimes to be more strict with her own work-life balance and and to prioritize uh, her hobbies and her things a bit more than she did sometimes in the past. But yeah, it's also brought me here, right? So
Yeah,
it's a process that I think teenagers have to go through.
yeah. Actually, that's an interesting perspective because while we can say I would change this or that, then maybe
Maybe I would be
the
different,
present
right?
would be very
Yeah,
different.
yeah. So it's now I look back and I think maybe I should have rested a bit more sometimes. But well, now it's too late. I don't know. So yeah, now I know for now I should rest more, do my hobbies and all of these. But I had to learn that. So
Of course.
I think it's it's maybe good if small Carolina goes through the same.
So maybe you would just tell her, oh no, carry on.
Yeah, I would just tell her, just have fun. Believe in yourself. Don't forget to have fun, and
I think this is
things
a
will be
great
fine.
this is a great advice actually. So thank you so much for
Thank
this
you.
conversation. And now we can move on to the Q&A. I would like to ask you then, you already said that you have many other interests, even as a scientist. So, what would you do if you were not a scientist?
Hmm. So I have a secret plan B.
Is it secret enough not to tell here?
Yeah, just maybe don't tell my supervisor. No, I had the pleasure to supervise a master student in my PG that she became a very good friend, and a very close friend. And when we were struggling in some days with some experiments, we had this crazy plan of opening a hostel uh, in Azores, so in a Portuguese island, where we would just do yoga retreats and hiking and diving experiences, and this would be our life. So maybe I would do that if I was
Maybe in
not
the
in
future,
science.
maybe in a parallel universe.
Maybe. Or having a small farm in the mountains could also be a good.
In the mountains here.
Yeah, there's no mountains in Portugal, so I
Yes.
guess.
Very good. Let's see, what else? Um, is there anything about your work that you would avoid completely? If you had, if you could say, okay, I will do everything except this, if you could remove one thing.
Uh, hmm, maybe cloning. So sometimes when we have to make a new, when we want to have a protein to be expressed, we have to do a process called cloning. Then it's very repetitive, and I get bored very easily, and I really don't like cloning. Every time I have to do some cloning, I have to breathe 10 times and really try to not postpone it forever. And yeah, because it's
Yeah,
just you pipette 4 tubes, you don't see any results coming.
one of those
It's
things that just have to be done.
exactly, it has to be done so that then you can go to the microscope and get good images. But if I could avoid all this cloning part and get already my protein to go to the microscope, I would definitely prefer that.
And what about when you were in high school? Was there any subject that you would have avoided if you could?
In high school, maybe English. I hated languages.
So, little did you know
But
that it
but
would
I
be your working language later on.
yeah, yeah. But it's still different that you like when you have to study a language or when you just yeah. I mean, of course, you have to study to be able to speak, but then you don't think about it when you are. I don't think about it when I'm working. Uh, but when I had to study for the tests, it was it was not so fun. In high school, I think that's it. All the rest, I quite liked it.
Okay, good. So, if you have any more questions, you can use these remaining moments of the episode to still post them. And besides that, if you have more questions in Portuguese or in English, you can send an email to Carolina. Her email will appear in our closing credits, and you can just contact her.
Mhm.
So thank you so much, Carolina, for accepting the challenge of being the first guest of this webcast, and for sharing with us how you became a developmental biologist. I have something for you.
Oh wow!
Just some Vista
Oh,
merchandise
thank you very much.
for you to take notes about your sea squirts. So, to our viewers, thank you so much for joining us today for the first episode and for the questions you posted. And don't forget to join us next week at the same time on the 11th of April for another Next Gen Scientist episode where you will learn all about becoming a glacier scientist. Until then, don't forget to like, share our videos, and follow us in our platforms. And when we publish this episode, you can also see the experiment that Carolina showed me. See you.
So this is how we keep our ascidians, our sea squirts. Their scientific name is Phallucia mamillata. And they look like this. They filter on the water, and they get fed on the water, and they have one siphon to feed and one siphon to secrete. And we keep them in these tanks, in these boxes where we can collect them. Okay, so we collect the sperm and the eggs in these tubes. This yellow thing is the eggs, and this whitish thing is the sperm. And now to fertilize the eggs, we have to activate the sperm. To do so, we dilute this concentrated sperm in seawater. And then to activate the sperm faster, we mix it to make sure everything gets homogeneous. And now we wait 1 minute and the sperm gets activated, so it's ready to fertilize the eggs. Okay, so now our eggs are ready. We can add the sperm, and we will add a lot of sperm so that all the eggs get fertilized more or less at the same time. So we add the sperm, we mix it to favor the contact between the sperm and the eggs. And you can see now, if you film the screen, you can see that now there's all this debris from the sperm when I mix it. This is because the sperm gets in aggregates. Now we can check if the sperm is active by checking with a different lighting, which is very hard to see on the screen, but I can see here that the sperm is moving around the eggs. And now we wait that they get fertilized. And now you see a lot of them got fertilized. So I will start washing so that we avoid more than one sperm entering the same egg and causing developmental defects. And now, because the eggs, after getting fertilized, they undergo some maturation and they have to start finishing their meiosis. You can see when this happens that they will extrude polar bodies, so some small cytoplasmic kind of protrusions that only contain DNA. And you can see that in some of them already being formed. For example, this one. This is now protruding a smaller polar body. And after about 20 minutes, we will have 2 polar bodies per egg. And we know that the fertilization was successful and they completed meiosis. And now you can very nicely see the first polar body. Before it was just starting to be formed. And now you can very nicely see, for example, in this one. Or for example, in this one, that you have this small protrusion coming out of the egg, and this will be the polar body. And if we look at, most of them have this, as you can see. So for example, these ones. So the fertilization was successful. Okay, so now we see that the embryos are starting to divide to 2-cell stage. So they are perfect for mounting for my experiment. We already prepared the dyes, you can see here. So we have one dye that labels the membrane, so this layer that involves each cell, and also the mitochondria. So it labels one of the organelles of the cell. And we prepare the dyes already in a mix. And now we will transfer our embryos that are ready to be seen on the microscope. We transfer them to the dye and we wait 3 minutes. So now I'm taking them. I move them to the dye. And now I wait about 3 minutes so that they incorporate the dyes, and then we can mount them for imaging. I already prepared the molds for imaging. As you can see, they have like this pattern. So we will put each embryo in one of these pockets, and then as soon as they are ready, we go to the microscope and we start imaging. When they are in these pockets, they don't move, so this allows us to see them with better resolution. Okay, so now they were incubating for 3 minutes, and now we can select some of them to put them for imaging in the microscope, and we just select 4 or 5. And now we put them in this grid system. Okay, yeah, so we have 4 embryos. They are already at 2-cell stage. And now we try to fit them into one of these pockets. And now they are ready for going to the microscope. Okay. So I found the embryos. Now I will put them, I will send them to the computer, the image, so that you can see. And here now we can select the one that looks better for imaging. So for example, this one. And now we can see here on the screen. Just reduce the resolution. So here we are labeling with this dye, we are labeling the plasma membrane. That's why you see this line surrounding each cell. And here we see just with normal light, like bright field. So our normal white light. And you can also see the pocket where they are, this well. And here we will see the mito tracker, so it labels the mitochondria. And you see also it's very punctate, so it's in these small dots everywhere in the cell. And now what we will do, in this microscope we can image each plane of the embryo. So we can define how much we want to image. For my experiments, I need to image the whole embryo. Okay, so now my image conditions are set. We are going to image the whole embryo, and we are going to leave it for about 2 hours. And we will just watch how the embryo develops, how the cell membranes look like, and how the mitochondria get distributed within the cells.
2 hours later.